Start the recording. And it is 10:41, so I'm going to say welcome to everyone. I'm Liskin Swint-Cruz, the Chair of Biochemistry and Molecular Biology, and we appreciate your coming to and participating in Hub. We hope next year we'll be all back in person like we have been in the past, but it's really nice to touch base with everybody, even if it's over this limited Zoom format. I am going to turn it over to our MC for introductions. Erin Fenton is our graduate director and has also been involved with Hub since the very beginning. So Erin?
Well, first off, I'd like to introduce this lectureship. So it's been named after Jerry Carlson. Jerry, would you say something so that your name kind of pops up at the top and everybody can see who you are?
Well, I'm just excited to hear Bob talk. I always like hearing Bob talk. I learn so much whenever Bob says anything. So.
Yes, that's true of all of us. So this is Jerry Carlson. He was our chair for many years and was exceptionally instrumental in getting the Hub Forum started. I want to say thank you to all of our guests for coming. We realize that this is not a substitute for being in person, and I don't think there's not a single person that's here that feels like this is a true substitute. It's kind of the best we can do. We definitely see things opening up and starting to see more things happen in person. So we are in high hopes that we get to be in person next year. But we wanted to make sure that our students in the area had an opportunity to present in some format as part of their education. So we did choose to have this as a online system this year. So our guest speaker is Robert Harris. We all know him as Bob. Bob has this very unassuming nature. He likes to be known as the farm kid that went into science, which is a background that I share with him. So we enjoy talking old farm stories, and we've even broke out pictures of our famed farm animals and swapped farm pictures. But if you step away from the kind of nonchalant approach that Bob has as a person, and that we all love having around for that type of attitude, the reality is he's a superstar. So he started his work training as a student in the Krebs lab. And when I say the Krebs lab, I do mean of the Krebs cycle, known as the TCA cycle. And his paper from the Krebs lab, actually Dr. Krebs thought he was doing Dr. Harris a favor, and Dr. Harris published his first paper as a single author paper. So he actually didn't publish with Krebs on the same papers, which now he kind of regrets just from the fame aspect. He actually has some background overlapping with Dr. Carlson in that they both went through Wisconsin, kind of at the height of the Enzyme Institute. And Bob has never relied on the fame of coming from the Krebs lab. In fact, much of the biochemistry in our textbooks, Bob has touched. So almost all the different regulations of glycolysis, when we think about coordinate regulation and glycolysis and gluconeogenesis, you get into maple urine disease and in the branch chain fatty acid metabolism, a whole bunch of that's Bob's work. He truly has just touched so many different areas. And then with his brother, Hank, he actually worked on some animal vaccinations, and they sold the first vaccination. The first one was largely driven by Hank's work, and Bob was more of a consultant, but they sold the first swine flu vaccination to Merck, and it's been in production more than 10 years. And I know that because after the 10-year agreement not to produce, they immediately started working on the next vaccination. And Bob is very active right now working on a vaccination for another type of swine disease. So he hasn't stopped. He's never stopped. Along the way, I believe it was 18 years, he was a chair of a department, biochemistry department at a medical center in Indiana. He has retired multiple times, and his last retirement brought him to Kansas City. And of course, he didn't stay retired long. He now has an office in our department, and it was a great privilege for me to get it published with one of my heroes. So Bob and I published a paper together. And again, he didn't stay retired. So he's now back on the vaccination trail working on the next vaccination. So Bob is truly a hero in science. He's one of my heroes, but he's a hero in the entire field. So I don't want to take more of his time, but Bob, we would love to hear your talk about your path in science and these great stories you have along the way.
Thank you, Aaron. I didn't want you to stop. I greatly enjoyed your introduction, although you stretched it here and there, but I really enjoyed. Thank you very much for that kind introduction. Please see my email address on my first slide. I'm hoping that I'll get some feedback, some questions, particularly from the students by email. Oh, Aaron, yeah, it's not changing. Here, we'll share a different way.
Get the mind.
No? Yeah, it goes. Yeah.
Arrows are working fine?
No. Yeah.
Okay.
Okay. All right, we're back in business. Sorry about that. We had it all set and then it didn't work, of course. When I sat down to think about the 50 years of working in research and teaching, I discovered there were a lot of people and that I've worked with, and it was very difficult not to just make it about those people because they have been so important with respect to my work. I have been blessed and very fortunate with having the opportunity to work with so many great mentors and graduate students and medical students and collaborators. I had a technician that was with me for 25 years, Pat Jenkins. I was really blessed. I give you a timeline. I was born in '39, but I won't start there. I'll start with polio, which had an influence on me. And some of you may remember polio. I'm certainly old enough to remember I had polio when I was 12 years old. My big fear was ending up in an iron lung. I didn't. I had problems with my legs, but I escaped the iron lung. My classmates, Frank Johnson and Robert Pearson, were not so lucky. Frank was permanently disfigured and Robert lost the use of his legs. My hero, of course, at that time was Jonas Salk, who, this was 52 when I had polio. That was at the height of that pandemic. And Salk came up with a vaccine in 54, and all my neighbors lined up to get the shot. And we got rid of polio. Some defining events in my life. I was raised on a farm, very active in 4-H. We lost the farm and we went broke, actually. We were too far in debt. We couldn't continue in 57. It was very painful, but it was a good thing, actually, because I changed high school and I was able to take chemistry. The high school I was going to didn't offer chemistry. And I fell in love with chemistry. And so that was really an important turning point for me. I enrolled in chemistry at Iowa State University in 1958. We still didn't have much money, and I was totally against going into debt. And so I got a job. I had a job all the way through undergraduate school. and I got a job in a foundry, Quinn's Wire and Iron Works. I started there just out of high school, worked the summer, and then when we went to school in the fall, I kept working in the foundry when I could find time, weekends, and also on vacations. I tell you something about that. In the mornings, we would make black sand molds for daisy hogwaters. That's what these molds look like. And in the afternoon, we got to pour the iron into the molds. And then when the iron cooled, we shook the hogwaters out and knocked the sand off of it. And I tell you all this because of impact that it had on me. You can see hands here. And if you work with this black sand, at least back at that time, they used a binder. It was an aromatic polyisocyanate. And that polyisocyanate reacted with hydroxyl groups and formed a polyurethane that held this sand together. But it also bonded covalently the sand to your hands. And then that became important to my career because I applied for a job at the Vet Med Research Institute. And I applied for a chore boy, which is something that I was certainly qualified for because I'd taken care of cattle and hogs all my life up to that point. And there was competition for the job, but the guy who interviewed me, Dr. Schweitzer, he asked me what I was doing, and I said I worked in a foundry and was going to school. And he said, "Let me see your hands." And so I showed him my hands, and he said, "Okay, you got the job." So then I was in the Vet Med Research Institute doing chores, taking care of cattle, hogs, guinea pigs, rats, so forth. And I got the opportunity to become a lab assistant in a biochemistry lab at the Vet Med Research Institute. And that happened again, just by sort of accident. They asked me if I would mind cleaning the toilets during the weekends when I was out there taking care of the animals. And of course I said, okay. And I found that the toilets were incredibly dirty, but they wouldn't come clean with normal cleaning solutions. And it was because it was rust. In other words, it was iron oxide because of the iron in the water. Well, I had to look it up, but I found that HCl would take the rust off, and because it converts it to ferric chloride, which is soluble. And I cleaned the toilets, and I cleaned them really well. And this biochemist who had a lab there, he wanted to know who did that. His name was David Gamble, and he found out it was me, and he talked to me, and he make a long story short, he offered me a position in his lab. And I worked with him then the next 3 years. I gave up working out the foundry. And actually, I got my first paper with David Gamble while I was an undergraduate student. And once I was with Gamble, I was hooked. I was hooked on biochemistry. And then I went to graduate school at Purdue. Undergraduate completed in '62, went to graduate school in Purdue and finished my master's and Ph.D. in '65. So my training in biochemistry then, I went to Purdue. I finished in '65. And I went then to the Enzyme Institute at the University of Wisconsin. But again, that was a strange story of how I ended up there. I went by way of Berkeley because, well, sort of. I did, in the last year of my PhD work, I applied for a fellowship from the Multiple Sclerosis Society for my postdoc work. And I got a job, I got the postdoc, I got the funding, and I was to go to work with Chekhov, I.M. Chekhov, who was a major person in cholesterol. I had worked on cholesterol for my PhD. And so he was willing to take me as a postdoc. And I was scheduled to start with him in January of '66. And on the day I was going to leave to drive the southern route from West Lafayette to San Francisco, Chekhov died suddenly and unexpectedly. And I was informed that everybody had to get out of Chekhov's lab. So I didn't go I ended up, and fortunately the multiple sclerosis people were kind and let me take my fellowship to the Enzyme Institute, where I worked then in David Green's laboratory. But I didn't work with him the first year. He assigned me to Takeaki Ozawa. Ozawa was a professor from Nagoya University. He was the chair of the second biochemistry department at Nagoya. And Green put me to work with him, and Ozawa couldn't speak English and I couldn't speak Japanese. But we learned to communicate, and I became his technician. I was a postdoc, but actually I worked side by side of him at the bench for a year as a lab tech, and that was a good thing because I learned a lot about mitochondria from Ozawa. And then Ozawa went back to Japan, and I just became a regular postdoc with David Green, and I worked in the area of bioenergetics until for the next 3 years. I'm going to show you this picture of a mitochondria just to tell you a story, and that was that while I was there, I worked with a pathologist, Junpei Asai from Nagoya, and he became professor of pathology. And Junpei noticed these ultrastructural changes or forms of mitochondria in heart mitochondria that I had isolated for him. And we figured it out, he and I, that there was a low energy state. When they were in a low energy state, then the matrix space was all collapsed. And then when I gave them substrate, to eat and burn, they went into what we called a high energy state. And so we came up with this hypothesis that electron transfer gives energy conserving conformational changes that resulted in configurational changes. It went from this sheet-like structure to these tubular-like structure. And that was energy conserving, and that would serve to make ATP. Well, we published that, and I wish we hadn't, but we did. And because about the time our paper come out, the chemiosmotic hypothesis came out by Peter Mitchell, and I'm sure a lot of you, or maybe most of you, have heard about or know the chemiosmotic hypothesis. And it says electron transport conserves energy in the form of membrane potential and pH gradient. And then that energy is used to drive ATP synthesis. And right away we said, oh my God, we get it. This is ion transport, it's causing an osmotic pressure change. That's what's responsible for our mitochondrial ultrastructural transformations. So we were wrong. And I bring this up because it was an important lesson. It was very painful. And I really seriously considered going back to the farm at that point because of the embarrassment of being so wrong. But on the other hand, it was a good lesson. And also, I learned that, that scientists make mistakes. And the important thing is that scientists often will admit that they make mistakes, and that's not true of politicians. They never admit a mistake, but scientists have to admit it, and it happens all the time. Of course, occasionally scientists are also politicians, of course. So anyway, in spite of my major error, David Green at the Biochemistry Department at IU School of Medicine decided to take a chance on me and hired me in 1970 as a faculty member. And I was there, I was in that position for 2 years. And during that time, I gave quite a few lectures. I gave my first lecturing. I went to a medical school because I didn't think I could give lectures and I didn't think I would be a good teacher. So I chose a medical school because I had heard the teaching load would be low. It was not so low. I immediately got involved with teaching. And I gave my first lecture in Emerson Hall Auditorium on September 24, 1970. And this hall is a big hall, and there was 300 and some students. It was combined medical students, dental students, and graduate students. And Gibson suggested I lecture on oxidative phosphorylation, which I did, my very first lecture. And before my lecture was over, it was raining airplanes from the balcony, which is another really major embarrassment. And I still had to give additional lectures. And you know, this was 1970. Everybody was pissed off at that time because of the Vietnam War, including me. And so it was a really tough time for students. It was a really tough time for faculty. But it was a terrible time to get up and give a research lecture to these students when that was definitely not what they needed, wanted. Anyway, that was my first experience at teaching. I got saved sort of because then I got an opportunity to go to Oxford to work in the Radcliffe Infirmary with Hans Krebs. And so I tell you how that happened, which was, again, so this is a picture of Hans Krebs. As Aaron mentioned, he got the Nobel Prize for the citric acid cycle. He came to Indianapolis for a symposium. I wasn't invited to the symposium, but I was there. I didn't even know about the symposium. But I got a call from a secretary and she said, Professor Krebs wants an audience with you. He wants you to bring a reprint of your halothane paper, a paper I had published just recently at that time. And I thought it was a joke. I could not believe it, and I didn't believe it. Dr. Gibson convinced me to follow it up, and I was really convinced that Krebs had found something wrong with my halothane paper. So I went totally prepared to defend my paper with him. And I met with him, and he asked me for the paper, and he asked me if he could keep it. So after we had a good talk, really about what I was going to do in the future, and I told him I had decided to work on lipogenesis, on the regulation of lipogenesis or lipogenesis. And he said, why don't you come to my lab? So I went, 73, 74, I was there for a year. And as Aaron said, I got one paper out of that work, and it's on the inhibition of fatty acid synthesis by cyclic AMP. And I wanted to publish with him, but he said he had enough papers and that he thought it would be good if I had a single author paper. And as a matter of fact, it's my only ever single author paper. So timeline of teaching, I taught in an introductory biochemistry course, intermediate metabolism, which is really good for me. I taught dental students for too long. I taught a lot of medical students for my medical biochemistry my entire career. I became graduate advisor, and I still taught those courses, except I dropped out of the dental program. And then I wrote chapters for the textbook of biochemistry with clinical correlations, one of them with my colleague, David Carab. And I became chair of the department in '88. And I didn't serve all the way to 2009. I've forgotten when I when I stopped. I became a regular faculty member before I retired in 2009. I retired in 2009, but I didn't really retire. I took my lab to the Roudebush VA Med Center. I was 70 at that point, but I got this job as a Deputy Associate Chief of Staff for Research. I kept teaching in the medical biochemistry course until I retired then from the VA in 2016. And I came here, and Jerry Carlson kindly gave me a position in the biochemistry department as a volunteer faculty member, which I greatly appreciate. Now, a word about teaching. I was blessed with a really wise mentor, Dave Gibson. Once I had that experience with the airplanes floating down on me while I was lecturing, I went to every lecture that Dave Gibson gave, and he came to a lot of my lectures and gave me a lot of feedback. I think the key for me, and probably for a lot of people, is empathy. I didn't start out with great empathy for the students I was trying, but I began to appreciate what they want to do. And so, I like this. I like this quote from Jim Henson. He says, "They don't remember what you try to teach them. They remember what you are." Oh. So my research direction really influenced by all these things. My 4 years in working with mitochondria with David Green, Oxford for a year with Krebs, and it seems strange now, but isolated liver hepatocytes were really enlarged. The procedure was largely perfected in Krebs' lab during the time that I was there. And we worked on gluconeogenesis and glycolysis. There were people working on that. I worked on lipogenesis, but there were people working on fatty acid oxidation and ketogenesis. And we had a meeting every Saturday morning, and sometimes it was hours long because it was, really great people would come and to participate. And I don't think I ever said one word in that meeting, but they were good. And of course, my medical lectures to the medical students had an enormous influence on me, because I became responsible for the metabolism and metabolic regulation and metabolic interrelationships between tissues and all the metabolic diseases that we were covering. So learning that stuff, I guess you learn it. You really learn it when you have to lecture on it. So a word about research from Abraham Lincoln. You can read it, but of course he wrote. He was very gifted with what he could write, and so I like this quote, and I think it explains why those of us in research often can't quit. So my primary research interest, as Aaron sort of mentioned, have run the gamut of metabolism, gluconeogenesis, glycolysis, synthesis of glycogen, the degradation of glycogen, and fatty acid oxidation, ketogenesis. But I also got very much involved in the regulation of branched chain amino acid oxidation, leucine, isoleucine, and valine. And I worked on that for a long time. And then after that, I might as well go to the related complex, the pyruvate dehydrogenase complex, which I did. So I just throw these slides in to say that this is what, as this comes from my textbook chapter that I wrote, but it's been sort of my guiding light, I suppose, for my research. The interest in all of these pathways and how they relate to each other and how they relate to the tissues of the body. And I'm fascinated by the fact that when you're well fed, when you've eaten a lot, your liver is glycogenic, it makes glycogen, it's glycolytic, it tears down glucose and it's lipogenic. And then the muscle is glycogenic to get rid of your blood glucose or store it. And then the adipose tissue is lipogenic and stores fat. And the brain, it has to have glucose all the time. But then when you fast, when you starve, the liver turns around completely what it's doing. And, the liver is now glycogenolytic, breaks down glycogen, makes glucose and ketogenic, and is proteolytic. And the muscle is proteolytic to release amino acids so you can make glucose from amino acids. And the adipose tissue releases fat and fatty acids and glycerol to make glucose and give you energy in the liver. So that whole thing has fascinated me, throughout my career, really stemming from my lectures on this sort of thing. And how the liver can do one thing in one part of the fast or the starve feed cycle and do a completely different thing and the other part of the starve feed cycle, and how that switch can be made, and what are the regulatory mechanisms for doing that. That's been of great interest to me and has driven my research program. And I extended, of course, to diabetes, in which your liver is stuck in the starved state. It does what it should be doing in the starved state, even though there's glucose and amino acids and fat coming in from your gut. And so, it's a fascinating thing and important, of course, in diabetes, that of trying to understand this dysfunctional regulation that we have because of the absence of insulin. And then a similar sort of situation happens when you have tumors that get large enough to have a major influence on the metabolism of the body. That's also been of great interest to me. So the timeline in my lab is really, I look at it in terms of the people that worked with me. And you will see, for example, on here that there's a guy by the name of David Crabb. David Crabb he started with me when he was a first-year medical student, and he became professor of medicine and biochemistry, and he became the chairman of medicine at IU School of Medicine. So he's retired from that position now, but you look on this, he's here throughout my career. And this is Yashi Shimomura, who after Ozawa went back to Japan, he invited me to come to Japan. So of course I went, and I was there for 4 months only, but I met this guy, Shimomura, and he was finishing his Ph.D. And Shimomura came to my lab during this period, but actually he just came and he'd stay a year or 6 months, and then he would go back and forth many, many times. And then I was lucky to have Bei Zong. Many of you may have heard of Bei Zong. Bei Zong was my PhD student, and she did wonderful work in my laboratory. And Bei, some of you may know, is now Vice President of Metabolic Research of Pfizer. I was lucky to get Kirill Popov to come to my lab. Kirill Popov came from Moscow University, and with him he brought the FBI. Well, the FBI followed him everywhere for a long time, but he came, no money in his pocket, but he came as a postdoc, and I was really, really lucky to have Kirill. And Kirill is now professor at Alabama, Birmingham. And then more recently, I've worked a lot with NQ Lee. He's professor of medicine and endocrinology at Koongpook University in Daegu, South Korea. So since my retirement, actually, I've worked a lot with NQ Lee and his people. So why interested in the regulation of the pyruvate dehydrogenase complex? This has been occupied a lot of my time and energy in the last, I don't know, 25 years. And it's the increased activity of pyruvate dehydrogenase lowers blood glucose levels. That's why we're interested in it. And it makes the kinases that regulate that enzyme potential targets for treatment of diabetes. That's what's driven my research. So I just give you introductory comments about the pyruvate dehydrogenase complex to maybe remind you or whatever. Pyruvate dehydrogenase takes pyruvate to acetyl-CoA, takes a 3-carbon pyruvate to acetyl-CoA, and it generates NADH. And it's regulated by phosphorylation. There's kinases that regulate it, and that's where a lot of my effort has been on those kinases. And there's phosphatases that remove the phosphate and activate the complex. In the fed state, the PDKs are downregulated, and that gives you less phosphorylated PDC, it gives you more PDC activity, and pyruvate dehydrogenase links glycolysis with the citric acid cycle. And so all the glucose you eat in a day has to go through this reaction. You have to get rid of all that glucose. You can't store it. You store it temporarily, but you can't store it forever. And so the glucose goes through this reaction. And so this is an important reaction for clearing glucose from our body. And in the fed state, the enzyme is very active, and you oxidize the glucose to CO2 and water. Or you can also take some of the acetyl-CoA and make fat out of it, of course. And so this enzyme is active when we're in the well-fed state. And then we starve. And in the starved state, you have to inactivate the pyruvate dehydrogenase kinase or complex. So the kinases are very active and the phosphatases are downregulated. And that conserves pyruvate for glucose synthesis. So a lot of gluconeogenic or glucogenic amino acids, lactate and pyruvate, alanine, and some other amino acids have to go through pyruvate to give you glucose. If the pyruvate went to acetyl-CoA, you couldn't make glucose out of it. So we shut down the pyruvate dehydrogenase complex, and we shut down the pyruvate dehydrogenase complex all over our body to conserve gluconeogenic amino acids and lactate and pyruvate for the synthesis of glucose when we're starving. It's essential for survival. So timeline of my research again, actually in these early days, we were working on lipogenesis. That's what I did in Krebs' lab, but then we continued that work, and David Crabb joined us, and we were working on dichloroacetate. And David Crabb came to my office, and we often chatted, and he said, you know, we know the pyruvate dehydrogenase complex is regulated by phosphorylation, but I think probably the branched chain ketoacid dehydrogenase, which is a enzyme for leucine, isoleucine, and valine metabolism, is probably also subject to regulation by phosphorylation. And him suggesting that set the tone for the next, I don't know, 40 years, okay? Because we started working on branched chain amino acids. We isolated the branched chain ketoacid dehydrogenase. We showed it was subject to phosphorylation and dephosphorylation. And then Shimomura was really good at isolating enzymes, and he isolated the kinase. And that was a major, because it's the abundance is very low. And then Bay came to my lab, and Bay and David decided we couldn't go on without learning molecular biology. I didn't know any molecular biology. They decided we needed to do it. So David and Bay learned molecular biology. They, I guess David went to Purdue for a while to learn molecular biology. I learned some molecular biology by osmosis from these guys, and we got into molecular cloning. And we cloned everything we could isolate. We were isolating enzymes like crazy and getting amino acid sequence, and then using the amino acid sequence to get the DNA. And we got into maple syrup urine disease. That's caused by eating too much maple syrup. No, it's not. maple syrup is urine disease. Your urine smells like maple syrup. And at Riley Hospital, which was right next to our lab, they had a patient, a Mennonite patient who had maple syrup urine disease. And they and David worked on and found the mutation that caused the maple syrup urine disease in the Mennonites. And that led to the elimination of this terrible disease in the Mennonites. And also at that time, there was we discovered, well, actually a veterinarian from Australia contacted me, and he said, you know, that we have polled Hereford cattle in Australia, and they have maple syrup urine disease, or they're carriers, and about a third of the calves that are born in Australia from polled Herefords never get up off the ground. So he came and he worked with Bay, and they found the mutation that caused maple syrup urine disease in those cattle, and that eliminated it then, or resulted in the elimination. Kirill Popov showed up, and he went to work with on the BDKs and the PD, and he cloned the PDKs. We thought there was one PDK, and he cloned PDK1, and then he found there was a second one, and then a third, and then a fourth. So there was PDK1, 2, 3, and 4. It was very exciting. And he also, we thought there was one phosphatase for regulating the PDH, and he cloned with Bo Li Hong, he cloned PDP1, and then discovered there was a second PDP, is PDP2. And then they cloned the branched chain ketoacid dehydrogenase phosphatase, and there was only one phosphatase then. That was a terrible problem because that enzyme had been isolated previously, the phosphatase, and we couldn't reproduce that work, and it turned out that work published by another laboratory was wrong. What else? We turned our attention then to regulation of the expression of these, and we found that, of course, in starvation, the PDK4 goes way up, and the PDK2 goes way down in starvation. And we looked at diabetes, and we looked in a number of different states to find the expression. And we looked at the promoter, Hai-Soo Kwon did promoter work on the PDK4. And we found that, you know, insulin regulated via the promoter inhibited the expression of PDK4, and found that cyclic AMP, glucagon, glucocorticoids, and so forth, of course, work through the promoter to promote the transcription of PDK4. And then we decided to make PDK4 knockout mice and PDK2 knockout mouse, and that was done by Nam Ho Jung. Oh, I forgot, but we also made PDK knockout mouse. We made PDK knockout mouse, and we found that mouse had epilepsy. And that turned out to be a mechanism, one mechanism that causes epilepsy. PDK deficiency in humans causes epilepsy, and that was only found after we found it in mice, or showed it in mice. But we made the PDK knockout mice because we thought that would show that it was a good, PDK4 was a good target for the treatment of diabetes. Nam Ho Jung came to my lab. He was sent there by NQ Lee, and Nam Ho was going to stay a couple years, and he ended up staying 9 years. Yasmin Rohimi from Afghanistan. They left Afghanistan after her father was killed by the Taliban. Yasmin worked on the PDK4 knockout mouse and did some nice work. And these people, Young Hoon and Themis and Yang Kyo and Bo Yoon, these are students that I worked with in Q Lee's lab. And more recently now, I'm working with Ben Wilbright and also Aaron. So I'll end up by just mentioning the last 3 papers I've worked on with people. This is Young Hoon Go. He really enjoys eating, and he would kill me if he knew I used this. But anyway, he did some work showing that inhibition of PDK2 protects against fatty liver by these mechanisms. And this is Byung-Kyu Min. Min showed PDK deficiency reduces inflammation by preventing polarization of macrophages to the M1 phenotype. PDK4 deficiency, PDK4 inhibition does a lot of things, huge number of things. We're still discovering all the things that PDK deficiency does. I don't know if it can ever be used as a drug target for diabetes, because it does so many things, like prevents macrophages by being converted to the M1 phenotype, which we need to do to fight bacteria. And then finally, this is Bo-Yoon Park's paper. She discovered a novel mechanism for lowering blood glucose. It turns out the PDK4 deficiency interferes with glucagon signaling, so it reduces glucose levels by that mechanism. Cheers. Thank you very much. I'm sorry if I ran over time. Cheers.
So our approach in this hub forum is we have questions. We'll first ask for any questions from our visiting students. And I know the Zoom interface tends to let people hide in the background and just stay hidden, but this is a great opportunity. So I encourage you to go ahead and ask any questions you might have. Okay, well, it will stay open to students, but we'll go ahead and expand the floor. And if any of our grad students have questions, please jump in. Okay, we'll open the floor to everybody because Jerry Carlson's sitting there jumping at the bit to ask a question, I can tell.
Sure, Bob, all those students are out there, and you've seen a lot of changes, and I certainly have. What advice would you give to the students at this stage of science, this stage of funding, et cetera?
Well, I suppose, you know, I did think about that a little bit, but what I was thinking more about, if you're undergraduate student at this point, I guess I would suggest, you know, you take a course in scientific writing, and that was very helpful to me. There was a scientific writing course at Iowa State University, and it was for engineers. They had never had a science major take it, or a chemistry major take it. And so the instructor thought that was terrific, because for the first time he had somebody in the course who wanted to take it. And so that was a valuable course to me, maybe because the instructor spent so much time with me. But anyway, that was important. I also think that recommend that people take physical chemistry. I apparently missed the lecture on osmotic pressure and when I was doing my research, but nevertheless, I think that the physical chemistry has served me well all my life. And people tend not to take it. So if they can get away with it, you know, but I recommend it highly. Those two things come to mind. And then more pertinent to your question, Jerry, is that I think that, you know, you have to be aware of where it's at, you know, where science is at. I think that, and you have to be really careful you don't get stuck. You don't get stuck and not be able to move with the times and be with it. And I think that, you know, for me, a good example was that, you know, I was trained as a chemist, and then I became a biochemist, I guess. And it was really important to move with the times. When molecular biology became a really important tool, a lot of my colleagues didn't make the transition to using it for their research. And so it was critically important that I did that. So I think that if I was now young and you know, considering a career in science, I would be really careful to see where things are at, get into a lab that's really doing cutting edge research, and try to figure out what their next big thing will be that will move things forward in the field. And I don't pretend to be an expert on doing that. But you know, that as Aaron mentioned, I'm really excited right now because I'm involved with making vaccines, and we're trying to be cutting edge. We're making of course mRNA-based viruses, or I mean vaccines, but we're making the self-amplifying RNA vaccines. These are more difficult vectors to make. They're more difficult vectors to deliver to the animal or to the human. But on the other hand, it's where it's at. It's where we have to be. So I'm not sure I can add any more than that, Jerry.
Thank you.
Yeah. I hope people will feel free to send me an email and to follow up with a comment, questions, anything. You know, I think I was pretty good in the lab, and as they say, you get promoted to your level of incompetence, which certainly happened to me. But you know, for a long time, about all I do is sit in my lab or sit in my office and talk to people. I guess that's why I can still, as long as I can still do that, I'll be very happy.
So we do have a next session, and since we had a much more truncated version in our timing for today, I don't want to go too far, but Bob, I wonder if you might talk a little bit about transitions, both undergrad to grad school and grad school, you know, maybe more postdoc to faculty, because each of those transitions have their kind of unique aspects to them. And do you have any insights, you know, kind of undergrad to grad school transition, different ways of thinking, and then again for trainees going into faculty positions?
Yeah, you know, those are really difficult times, you know, to make those transitions, and you know, and it's really, I'd like to say that, you know, that I made those decisions and then never looked back. but it, you know, that's impossible, you know, and I guess I feel, I guess I sort of feel like Yogi Berra, you know, when you come to a fork in the road, you take it, right? And I can tell you that those are really difficult, and I don't have good insight with respect to you make those decisions on the basis of things that you just have to take. You just have to get to the fork in the road and take it. And I look at all of those forks in the road, and I try to understand why I took a particular. For example, you know, I faced a fork in the road when I got done with my undergraduate training, and and this name won't mean a lot to a lot of people, but it means a lot to some of us. And I got an interview with Linenger at Johns Hopkins, and I went for that interview, and I met with him, And you know, they put me up in a hotel in Baltimore, and it had an iron fence running around it with dogs, and when they told you don't leave your room during the night. And so they came and got me and took me to the interview with Linenger, and then took me back, and then took me to the airport. And of course, you know, I should have gone work with Linenger because he was famous man. And people are saying, oh my God, he could have worked with Linenger, but I couldn't do that. Farm boy going to Baltimore and have to be in a hotel with an iron fence with dogs running around, it was not, wouldn't work. So I made the decision, you know, to go to Purdue. I don't really regret it. I learned a lot at Purdue, but I think that, you know, it would have been something to got your degree with. You know, so, you know, I got a chance to, when I finished my postdoc, I had a good offer from California School at Santa Cruz. And people say, oh my God, why didn't you go to Santa Cruz? And, you know, and I went to Indianapolis. So, you know, those are really tough decisions. And you gotta, I don't know. I'm no good at telling you or answering your question because, you know, I came to these crossroads and I just took it.
Well, I think that in itself is a pretty good answer, just because there's never any two universes that you can live in. So
Yeah.
I think I don't think it's even healthy to second guess. You choose one and you go forward.
Yeah.
We're almost out of time, but Dr. Parnell has his hand raised. Do you have a question, Stephen?
Yes, Bob, I've seen many of your lectures, and I don't think I've ever heard you give a talk that you haven't ended with your catchphrase of cheers. When did that start? How long have you been doing that? And how did it start?
I think it started because when I was lecturing to the medical student, I know it started when I started lecturing to the medical student, and I had been involved in hiring a guy from England, who some of you know from Scotland, actually, Peter Roach, and you know, would always often drink beer or wine with Peter, and always he would say cheers, and it sort of stuck with me, I guess. And so I started saying cheers after all my lectures to the medical students, and it sort of I think it originated with Peter Roach. Yeah.
All right, so I think we're ready to move to the next session, which is a different Zoom link. And I'll just say that I've had several students ask me, when did I think of myself as starting to be a scientist? And I actually have answered, I don't know, because it's really kind of challenging to answer when you start being the scientist. And I think the early stages is the conversations as a student. So I encourage you to think of yourself, even if you're the undergrad student, to think of yourself as the scientist, and as scientists we communicate. So Bob gave his email address, and I encourage you to continue the conversation. And if that includes questions for Bob, please send those. So I look forward to seeing presentations from our guests in the next session.
Yeah, I'm just going to give a few procedurals here. Everybody will go on and switch over. Aaron, if you will go ahead and switch over, I believe you'll be able to open up the breakout room as co-host. I'm going to stop the recording, and it will take it a few minutes to save. So it will take me a little bit to get there.
Okay, I will exit now.
And the rest of you go ahead and exit, and I will see you over there.